Okay, before we get into Chapter 11, let's do a little review from Chapter 10. So for our first question here, endospore-forming bacilli with gram-positive cell walls are in which group of bacteria? Is it A, the actinobacteria, B, the proteobacteria, C, the chlamydias, or D, the firmicutes? Go ahead and pause the video, think about this one. All right, the name gives it away here. Endospore forming, firmicutes, which means tough skin. Endospores are those tough skins, so it would be D, the firmicutes here. Okay, let's do one more. You discover a new bacterial species with a very unique phenotype. It can produce oxygen. Based on your knowledge of the main bacteria phyla, which phyla would you predict that the newly discovered bacteria belongs to? Is it A, the actinobacteria, B, the proteobacteria, C, the cyanobacteria, or D, the firmicutes? Go ahead and pause the video, think about this one. All right, the answer here would be C, the cyanobacteria. Those are our main oxygen-producing photosynthesizers. Okay, so hopefully those questions weren't too challenging. Some of the test questions will be much like that. It's difficult to get into much more detail because we just did a brief overview. In Chapter 11, we are going to do a similar overview, but this time we're going to look at eukaryotic microbes, as well as some things that aren't technically microbes, but are included in microbiology for a couple of reasons. These chapters, the sections are going to be focused on Chapter 11.1, 11.3, and 11.5. There is interesting information in the other sections, fungi and algae, but we don't have time to go into it. Most of these eukaryotic disease-causing organisms we call parasites. So that is kind of the way they're used medically and scientifically. When we're talking about a parasite, we tend to be referring to a eukaryotic microbe. So let's start with a case study. We have Mario. He is a pizza delivery worker in Indianapolis. He's 29, and he has this large open sore on his arm that wouldn't heal. The sores develop into these red depressions in the middle, and they have kind of a raised border around them. I'll show a picture in a moment. It doesn't really hurt, so he's busy and he doesn't go seek treatment, which is something I think we can all understand, right? There's a lot of things going on in life. Sometimes if it's not, if it doesn't feel like it's going to kill us, we don't go and see the doctor. So here's his sores, right? These are pretty gnarly looking, raised border, deep kind of craterous wound here. They started to heal, but after 2 months, they start to come back again. And this time they're coming back both on his skin and in his mouth. Now he's getting some other symptoms. He has fever, swollen lymph nodes, and weight loss occurring. So he finally goes to a clinic and reports his symptoms to a physician assistant. PA asks, has he ever been out of the country? Because this does not look like something that we normally see in the United States. He says 6 months before he came back from his tour in Iraq, he was in the army. And he, when the PA looks at this, they notice that the sores look like cutaneous leishmaniasis. This is a disease that is common in many kind of tropical and subtropical regions, but not in the United States. That's why they asked, have you been out of the country? They're going to do microscopy, which we will find is very important for diagnosing eukaryotic parasites. Because they're much bigger, we can see them much easier under the microscope, as well as a PCR to test for the presence of the protozoan Leishmania donovani. This parasite has commonly been acquired by soldiers in Iraq, and it is transmitted by these sand flies. So we will notice that there are several of these eukaryotic parasites that are transmitted by what we call insect vectors or arthropod vectors. These are things like flies that take blood meals from organisms, and in the process, they often tend to move these parasites from one individual to another. So here's a micrograph of Leishmania. They're single-celled eukaryotic parasites. They have these little flagella that move in a whip-like manner instead of the corkscrew-like bacteria. These like spade-like heads in there, and you can see there's a nucleus in there. These are much bigger than bacteria, so we can often identify them very easily with microscopy. Now, they prescribe sodium stibogluconate, which is also called pentostam. It's a mineral that contains antimony, which is an element in it which kills the parasite. This treatment, like many treatments, we don't fully understand why it works. This is still an active area of research. We don't understand the actual molecular mechanism, as it's called, that kills the microbe. So with a lot of these eukaryotic parasites, there are limited numbers of treatments. And we will talk about this repeatedly, but these are eukaryotes. You are also a eukaryote. So the amount of differences between you and the microbe is less than like you and bacteria. So anything that's going to kill the parasite might also kill you or have effects on you. So there are a number of side effects that are associated with this treatment. Being tired, loss of appetite, nausea, irregular heartbeat, even pancreatitis, which is inflammation of the pancreas, and it can be toxic in the veins. So this is not something that you really want to have to take, but in this case, he has this severe infection and needs some sort of treatment. We will see that side effects are larger in the drugs that are used to treat eukaryotic parasites. So his sores start to heal, but he unfortunately might not ever be completely cured or have the parasite completely eradicated. In the case of Leishmania, symptoms can reoccur months, years later. These parasites are really hard to eradicate. So for this reason, all U.S. military personnel who have been deployed overseas are restricted from donating blood for at least 1 year from when they get back, because they could have these parasites floating around in their bloodstream. This particular species, it's a single-celled eukaryotic parasite, like I said, commonly found in tropical and subtropical regions. Many of these diseases are tropical diseases, but there are some in our own area. We'll talk about Giardia later. This parasite can have different forms. He had a cutaneous form, which is a skin infection, which is generally not life-threatening. But there can be mucocutaneous, which is infecting the mucous membranes, and even visceral, which can infect things like the internal organs, the liver and the spleen particularly, and these forms can be very serious and can even be fatal. There's about 6 million people currently infected, with about 1 million new cases per year. One common factor of many of our eukaryotic parasites is they have relatively complex life cycles. This one is simple. It might not look that way, but we'll look at life cycles from things like malaria, and they are very, very complex. So we have the parasites, right? And we have them in a sandfly. It takes a blood meal from a human, and it has those parasites. They move through the proboscis into the bloodstream. We're not going to worry about the terminology here. There's a large number of different terms for parasites, but these promastigotes, they get pulled in by macrophages, but they start to multiply in host cells, and they avoid the immune system and start to multiply. They switch to a different life stage called amastigotes, which multiply and damage tissues, which cause the sores. Here's a particularly gnarly example of it. Then, like a sandfly might take a blood meal from an infected person, and they move through the bloodstream and get taken up by the sandfly. In there, they develop back into the promastigote form, and divide inside of the fly. It goes back, takes another blood meal from a different individual. The cycle completes itself. So this is pretty typical of our eukaryotic parasites. We will see a number of diverse different parasites in this chapter. So we're first going to do some review / a little more in-depth overview of eukaryotes. We need to talk about two processes, asexual reproduction and sexual reproduction, that are key to how eukaryotes divide and reproduce. We'll talk briefly about the fungi, algae, amebas, ciliates, and trypanosomes, and a few other things as well. We will talk about some of the things that are not microbes, but get lumped into microbiology because of some of the processes they're involved in. Okay, so a bit of review, right? Looking at the structure of the eukaryotic cell. Remember, the big defining feature is the nucleus, which contains the DNA in a membrane-bound sac. We have membrane organelles inside of the cell, the endoplasmic reticulum that helps where proteins are made, ribosomes are on there. The Golgi is kind of the packaging and shipping facility, and then vacuoles are little membrane-bound sacs that kind of move around the cell. And there's some specific ones like peroxisomes and lysosomes that break things down. We also have mitochondria. Those are the powerhouse of the cell, right? They produce energy through making ATP, as we've seen in the metabolism chapter. Plants and some photosynthetic algae contain chloroplasts, which are a different organelle that can undergo photosynthesis and produce oxygen in the process, as well as sugars. We're going to talk about two new terms here. This term ploidy and what it means. So we've been talking primarily about bacteria. Bacteria have a single circular chromosome. On the other hand, many eukaryotes are what are called diploids. They have two ploidy copies of the genome. So diploid means two copies of the genome. Each cell has two copies of the genome, and these copies can be slightly different because one of them comes from the maternal side and the other comes from the paternal side during sexual reproduction. This leads to what we call genetic variability, and it is one of the key factors of sexual reproduction. So we will talk about this in a moment. The other side of this is the term haploid, which technically means half, but for our purposes means a cell that carries one copy of the genome. In the case of animals, these would be the gametes, the sperm and the egg. So in sexual reproduction, these haploid sperm and egg, 1 + 1 = 2. They form a diploid zygote, and then that zygote divides and becomes the full multicellular organism we know. So let's look at the cycle here. We're going to focus on animals. This doesn't fit for everything, but I want you to know these terms. So we're gonna talk about these different types of division in a second. So we have you, me, everyone that is human. We are what's called a multicellular diploid. That means we're made up of many cells. Each of them is diploid, 2N. We have 23 different types of chromosomes, and we have two copies of each, which means we have 46 total chromosomes. In the ovaries and the testes, we have a process called meiosis that is undergone, and that divides the ploidy in half. That goes from 2N cells and makes 1N eggs or sperm. These eggs and sperm cells cannot live on their own, right? They're unicellular. They will fuse together in the process of fertilization. 1N + 1N = 2N. So the zygote, the structure that forms from sperm and egg fusing, is now diploid again. This is genetic variability, right? You get part of your genome, one copy from the maternal side and one copy from the paternal side, and those copies can be slightly different. That's Maybe one parent has blue eyes and one parent has brown eyes or something like that, right? They're giving the genes for those different things. And there's complex rules of genetics as to how that gets expressed. That is beyond this course, but it is fascinating, I guarantee you. This diploid zygote undergoes divisions called mitosis, which develops into all the different cells in your multicellular body. And then the process of meiosis occurs. We go back to one end, the cycle continues. We are going to talk about each of these divisions, mitosis and meiosis, because they are critical. Mitosis is the process of segregating the 2 copies of all chromosomes evenly into each daughter cell. So we start with 1 cell and we end up with 2 daughter cells with 2 copies of the genome. This is somewhat similar to binary fission, although we don't call it that because binary fission is for bacteria. This undergoes a different process than binary fission. Replication occurs, and the way this happens is very different than in bacteria. But you can think of it essentially the same way. They are clones of the previous cell, except in eukaryotes, we tend to have differentiation that occurs where cells become different after this division. Like they might divide into become something more specialized, like a nerve cell or something. So we could have like stem cells that become more specialized cells. Some eukaryotic microbes, like yeasts, can actually live indefinitely through this process of mitosis. They do asexual reproduction. This can be called vegetative reproduction. They don't have mixing of sperm and egg. They just divide by mitosis. That is how they reproduce. This is also how all of the non-gamete cells in your body are formed. If we were to break this down into kind of a mathematical equation, right? Mitosis is we start with 1 2N cell. So that's this, right? It's a single 2N cell. It has 2 copies of each chromosome in its nucleus. That 1 2N cell goes through mitotic division. It has to duplicate its genome. So essentially, the cell has 4 copies of the genome. Those get split apart in this process, and then we end up with 2 identical 2N cells, right? So DNA replication occurs, we split that apart, we end with 2 2N cells. We started with 1 2N cell, we end with 2 2N cells. Okay, I think that's pretty simple to understand. Please don't worry about the details of this. You'll probably go into it in an anatomy course because it is a little more anatomy focused. In the case of sexual reproduction, we have a problem. If you take a 2N cell and fuse it with another 2N cell, you would end up with a 4N cell. So as life progressed, our genome would get bigger and bigger and bigger because we'd have more copies in every single cell. So before sexual reproduction can occur, we have to half the ploidy number. We have to divide the genome in half, essentially. And this process is called meiosis. This is a specialized cell division that forms gametes in animals, at least. Plants and fungi are different. We're not going to get into that. So the end result is going to be 1N cells that can then combine together to form diploid cells again. So 1N plus 1N equals 2N. This increases genetic variability because we're getting a little bit from the mother, a little bit from the father, and it's mixing. And there's also the process of recombination that happens in here. Recombination is mixing of maternal and paternal DNA that your sperm and egg cells have. So that is beyond us. Again, you can take a genetics course if you're interested in that. So what happens in meiosis? Well, meiosis starts out like mitosis. We double the genome, okay? So we take a 2N cell and we actually make it into a 4N cell. In the process, some genetic recombination can occur, but then we split that in half. So we have 2 2N cells. But then a second division happens with no duplicating of the genome. This divides that in half, so we end up with 4 1N gamete cells at the end of this. So we start with 1 2N cell here, and we end up with 4 1N gametes at the end. Those could be sperm or egg. That is the result of a single meiotic division. So that is how this cycle works, right? Meiosis divides the ploidy in half. You get sperm and egg. Fertilization combines 1N plus 1N equals 2N. Mitosis occurs. All of your cells grow and divide, and then the cycle continues. I cover those divisions because they are key to how these microbes can reproduce. There are sexual stages to many microbes that are eukaryotic, and also it increases their genetic variability, so they have ways that they can evolve rapidly because of this mixing of genetic material from sexual reproduction. We have a variety of different categories of eukaryotic microbes. There are microscopic fungi and microsporidians, things like yeasts. We aren't going to talk about them too much because they are very diverse. There are several that can cause diseases, but we're not going to get into the fungi too much. That is beyond us. We have the amebas, single-celled, what are called protists. I'm going to use this term protist. Protist is it's an old term that was kind of used historically to lump together all the single cellular eukaryotes that we didn't know where they fit into the tree of life. Through DNA technologies, we have found that a lot of protists are not, in fact, evolutionarily very closely related to each other, but in the past they were lumped together, so we kind of talk about them together as well. We have amoebas in there. You've probably seen, and we talked a bit about them, phagocytosing. Ciliates, we're not going to talk too much about them. Apicomplexans, this is an important group of intracellular parasites. That means they live inside of cells. They have this special apical complex that enables them to get inside of cells, which allows them to hide from the host immune system. We'll also talk about trypanosomes and metamonads. These are extracellular parasites. They live outside of our cells, and they often have complex life cycles as well. The algae we won't talk about too much, but they are important photosynthesizers. There are some, like dinoflagellates, that can produce neurotoxins. We talked about red tide previously. Then we get into the two groups that technically aren't microscopic, so they're not microbes, but they are included in microbiology. We include the worms because they have microscopic egg stages, and we include arthropods like insects because they are often vectors of these other microbial diseases, so they transmit them. We talked about fleas with the plague, and we talked about the sandfly just in this chapter, actually, and our leishmania. So the insects are key in moving pathogens and parasites from one organism to another. We call that a vector. So that's why they're included in this. The worms are the creepiest ones. They're gross. We'll talk about them. You might not think it, but a large portion of the world population is affected by worms, particularly in tropical areas. 25% of the world's population, maybe up to 30%, have some sort of worm infection. And it's hard to get reliable estimates for the U.S. I looked into this. Our book says 10%. That might be a little high, but it could be as much as 5%. Often in the southern regions of the United States, where it doesn't get quite as cold in the winter, these helminth infections, as they're called, are quite common. Okay, that is it for 11.1, our overview. We will go into two of our sections, 11.3 and 11.5, and talk about some important eukaryotic microbes.